Okay, if I can introduce Dr. Klára Panzarová from PSI, who is going to start us off on session 2. Thank you.
Okay, great. Thank you. And hello, everybody. I hope everyone is still fresh and full of power for the second part of today's afternoon. And I will follow up what Jeremy and Tracy introduced very nicely. I think there will be a bit of slight repetitions, but that's probably useful sometimes. So a very brief outline of my talk. So not much to be said about the measurement techniques because a lot was said, but I will introduce briefly the fast and pump fluorescent techniques. And then I would like to speak about the tools that can be used for measuring chlorophyll fluorescence. And finally, I'd like to mention a few details about the practical aspects of the measurement. And the last part of my talk will be about the applications. So I think we heard enough about what chlorophyll fluorescence measurement is actually, and very nicely being introduced. What I would like to maybe remind and mention again, that with chlorophyll fluorescence measurement technique, we get very detailed information about the photosynthetic performance of the plants, which is giving us very detailed or very good information about the, let's say, plant physiological status of the plants. And this in respect to growth conditions, to the developmental stage of the plants, also to stages of, let's say, stress responses of the plants, which can be the onset of the stress response, progression, recovery. And another important point to mention is that PS2 activity is very sensitive to range of biotic and abiotic factors. So chlorophyll fluorescence can be used as a rapid and sensitive indicator of the plant performance status. So basically what we get is, let's say, high resolution dynamic information about the plant status, which is very useful, especially as it's a rapid indicator. So usually with chlorophyll fluorescence measurements, we can actually characterize the stress status of the plants prior actually any stress effects are seen, for example, by visible imaging. So range of applications, and I will in the end of my talk introduce some nice examples. So we saw in Tracy's talk about example of screening and characterization, screening to, for example, different treatments. But it's a very useful tool for genetic variations, for ecological diversity screening as a stress indicator, this we mentioned. And lately, and very important to say, and in the next talk we will hear this from Kerstin, very important tool in crop improvement research. So I will not go into detail anymore because I think we heard very well about the fluorescence yield and how we can actually understand the different states of the fluorescence decay. So basically with chlorophyll fluorescence analysis, I think we can say that we can very effectively separate the components that make up for all the energy dissipation. And I will talk mainly about the tools that we can use for characterizing the individual components. And it's a little bit simplified, but basically I would differentiate the chlorophyll measuring techniques into non-modulated fluorescence techniques, which are techniques that are primarily referring to OGIP type of measurement techniques, fast measurements of fluorescence inductions in continuous and saturating light pulse, techniques that require dark adaptation. And this will be in the next few slides I will mention briefly, and mostly I will talk about the pulse amplitude modulated technique, which is based on using rapid pulsing light source measuring flashes that specifically induce fluorescent signal from the sample. The key feature of this technique is using sophisticated electronics, which allows actually to discriminate the fluorescent signal from the measuring light, from the ambient other light. So basically by ignoring any non-pulsed signal. And due to the pulse amplitude modulated technique, we can also do all the measurements in the light environment as in the field conditions during the full sunlight. So in the next few slides, I'd like to briefly mention the non-modulated chlorophyll fluorescence measurements, so-called primarily OGIP type of measurements that probably quite some of you might know. And basically what we do is we measure a polyphasic rise of chlorophyll fluorescence from F0 to Fm under a strong excitation pulse. The illumination usually ranges in about a second. And for measuring this OGIP, OJIP type of curve, basically the key requirement is using very high resolution, time resolution detectors. So basically measuring in 10 microsecond scale, especially in the very first phase of the measurement. So the OJIP is usually plotted on the logarithmic scale, and this is especially due to the rate of the measurement in the very first phase, which has to be really on the level of microseconds. The result of OJIP type of measurements, a range of parameters that can be extracted that are characterizing the OJIP curve and different phases of the curve. In summary, very complex. You see, it's a dramatic list of parameters, not all so easily transferable into the practical applications, but parameters that refer to energy fluxes in the plants and the quantum yields and efficiencies and specific energy fluxes, for example. I would like to mention briefly two examples. So basically, one more time, what we measure is the rise from the basic dark adapted fluorescence yield to the maximum yield, so the OJIP, transient state. And primarily what we get is, or what we analyze is the changes or the status of the electron flow around the photosystem too. And here, one example that I would like to share, application for the OJIP curve, and that's abiotic stress type of treatment where plants were treated with ozone, and we can see that basically fairly fast after the treatment with the ozone, we can see a drop in the maximum level of fluorescence and quite tremendous change in the type or the shape of the OJIP curve. And one example how OJIP can be applied in the studies where early symptoms of the biotic stress plant response were captured, and work from the group of Thomas Reusch. And basically what you can see here are Arabidopsis plants that were infected with virulent and avirulent strains. And by using the OJIP curve and by applying novel algorithms for the identification of a unique fluorescent signatures, what the authors can show that basically they were able, based on the parameters extracted from the OJIP curve, discriminate between specific signatures for virulent strain and avirulent strain, which has quite an interesting application. So this would be a brief introduction mentioning about the fast fluorescence kinetics type of measurement and the applications. And I would step now more into the pulse amplitude modulated chlorophyll fluorescence measurement that you heard about quite a bit in Tracy's talk. She introduced pretty well. So for this type of a measurement, what is essential is the fact that the fluorescence is excited by the short flashes of measuring light, which allows dynamically discriminate from the fluorescence excited by the continuous light or from any potential background light as ambient light we have, for example, in the field when we do the field type of measurements. For this type of measurements, we can use different we measure the photosynthetic system in different states, which I try to summarize here. So I think was introduced quite well that we can't tell much about the state of the photosynthetic apparatus from steady state fluorescent signals. So we measure the fluorescent yield in, or we measure the photosynthetic system in known states. And this is by applying different types of light sources for the measurement. Weak light, which is the measuring the light, the pulsed light. Strong light when we measure the maximum fluorescence levels, and then actinic light where we can measure the effective photosynthesis. And I would like to talk a little bit more about this in the few slides. I'm just checking how far I am with the time. And so the key, so basically speaking about the 3 roles of the light sources in plant fluorescence experiments. And the first is the measuring light. And with the measuring light, we excite the fluorescent emitting pigments without changing actually photochemically active state of the organism. And this means that we basically measure the minimum level of fluorescence by keeping the photosynthetic organism in its maximum effectivity. So the measuring light is usually typically 10 to 30 microseconds long flashes with precisely repeated low frequency of applications. The key point is that the rate and intensity of the measuring light is not causing any actinic effect. Actinic effect here means that basically the intensity of the measuring light is so low that it's not inducing electron transport through photosystem 2. The intensity of the measuring light is usually around 0.2, 0.5 maximum micromole per square meter per second. So this would be the measuring light for measuring the minimum level of fluorescence. The second role of the light in the measurements, or the second type of light we use, are actinic lights. The actinic light excites the chlorophyll fluorescence molecule into the frequency that is comparable to outer conditions or to ambient natural sunlight conditions, resulting in basically driving the photosynthesis. So resulting in or bringing the photosynthetic apparatus into the status when the substantial photochemical activity is generated. And generating so-called instantaneous fluorescence level that we can easily determine and measure. So the actinic light is typically continuous light, continuous light of defined time or length and intensity as well. Usually it's a moderate power and usually refers to cultivation light intensity when speaking about the intensity of the actinic light that one should use. So it's the light that drives photochemistry and photosynthesis. And the third light that plays a key role in chlorophyll fluorescence measurements is the saturating light. And this is the light that excites the pigment molecules into the capacity that exceeds actually the capacity of the photosynthetic apparatus. So with the saturating pulse, which is usually around 800 milliseconds, 1 second, no photosynthesis is operational. So photosynthesis is 0, whereas the fluorescence level that we measure is the maximum level of fluorescence. The irradiance of the saturating pulse ranges from, I would say, 2,000 to 15,000 micromole. In the imaging systems, it's usually at least 6,000 micromole per square meter per second. So having these 3 light sources, we have all what we need actually to measure effectively the fluorescence yield. And I will not go, I think, again in detail over the fluorescence yield curve and the application of the different light sources and the resulting states and the fluorescent transient states. Overall, what we measure and obtain is the amount of the energy that is being quenched by the photosynthetic organism, and we heard enough about the discrimination, the key discrimination of the processes that are referred to photochemical quenching or photochemistry and to non-photochemical quenching of fluorescence. So result of the quenching analysis, depending on the specific definition of the protocol, is a large list of measured parameters and list of parameters that can be calculated. So, and how can we measure chlorophyll fluorescence and what are the types of tools we can use? So briefly introduced by Jeremy, I will focus on non-imaging and imaging fluorimeters. I will just briefly mention. Basically, both types of solutions are fluorimeters which integrate the given 3 light sources for the photosynthetic measurement. Non-imaging fluorimeters, I just tried to pick up some representative examples. So the more complex type of solutions that integrate both chlorophyll fluorescence as well as gas exchange type of measurements are, for example, tools from Lycor and Waltz. There are some tools by Lycor that also integrate a porometer function into this type of measurement. Let's say more simple type of solutions that specifically focus on chlorophyll fluorescence or solely are designed to measure chlorophyll fluorescence. Kinetics are different versions of so-called fluorpen tools that are manufactured by PSI. And here solutions are available with different types of clips, depends on the type of application the user wants to use. And also solutions where simply one time point measurement can be acquired, or we can have long-lasting monitoring devices or long-lasting dynamic type of measurements in the field where the tools are installed in the field. That can be extreme type of environment as here in Antarctica, and or tools that are optimized, let's say, for underwater type of measurements. And the applications are broad for this type of simple measurements, especially when speaking about the fluorpen type of measurements. So we can very rapidly, effectively measure the photosynthetic parameters. Usually the tools are coupled with other type of measurements as GPS coordination or photosynthetically active radiation type of measurements. Plus, often they integrate also the fast induction kinetic measurements. And yeah, as said, I mean, the applications are very broad. Here, I would mention that actually the use of these simple handheld tools is implemented also in already, let's say, physiological breeding type of strategies, and was, for example, introduced different types of tools and the guidelines how to use them in the field guide to wheat phenotyping that was published by CIMIT, where all the protocols and optimizations for the field phenotyping are mentioned. So there I would recommend, in case, to have a look if someone goes and measures directly in the field. So this would be the type of applications for this simple handheld type of tools. And I would step and briefly introduce the imaging fluorometers and introduce the strategy used in the imaging fluorometers. And I think was nicely mentioned by Tracy, what are the pros of the approach for imaging type of solutions, as we obtain the entire spatial heterogeneity information for the plants. And in respect of the tools that are available, I mean, there are different versions of the imaging pumps from bulbs, and I would say quite a big plethora of solutions of fluorocam images that are manufactured and developed by PSI. And I would say today we can really find solutions which go over the entire scale of resolution, where we really can go from single cell type of measurements, where we can measure, it's shown by Tracy, basically on single cell level, over single leaf level, single plant level, up to actually populations, we could say, of plants. And this is mainly used in, let's say, the automated high throughput type of systems, where large panels are used for the sample illumination and allowed to screen, let's say, in really high throughput, high precision populations of plants for chlorophyll photosynthetic characteristics. And I would like to briefly describe actually how chlorophyll fluorescence measurement is performed, let's say, from a little bit more technical point of view. So what we obtain and measure are fluorescent transient states during a specific light illumination. And here we have an example of actually illumination curve, where we have measuring light minimum, measuring light for measurement of 0, so minimum level of fluorescence. We apply saturating pulse, then we switch on the actinic light, and we apply multiple saturating pulse during this light phase. So that would be like a scheme for the light stimulation. And in parallel, in the same moment, so fully synchronized, we acquire the fluorescent transient signal for individual pixels that are being imaged. And as a result, basically what we get is a fluorescent transient video sequence, or fluorescent transient states that are captured in the video sequence. So we have the time, and then we have the individual transient images being acquired in this video sequence. And as we know in which part of the sequence which light source is applied, we can select directly frames that represent, let's say, low contrast type of fluorescent signal, which would refer to F0 value, and usually we apply multiple of the frames together and average them. The same we can do for the high contrasting frames, which would be here, it's example frame 15 up to, let's say, something frame 20, And again, these frames for the high contrast are used for actually averaging and obtaining the FM image of the plants. So when this is taken together, basically what we get is pixel to pixel kinetic information, depending on the type of measurement we design, which provides us the spatial heterogeneity information over the entire surface of the plant. And we can do actually the pixel to pixel arithmetic calculations on the images that we obtain, resulting in obtaining the measured parameters and also the calculated parameters with all the spatial heterogeneity information that we can obtain. And what type of parameters we obtain depends on the type of protocol the users select or use. And I think this was quite well introduced in the previous talk about the different parameters we can use. I would like to, let's say, remind this and speak about the types of measurements from a little bit different of aspect. Now, let's say, speaking about more the practical aspects. And especially, I would use a little bit of experience we have from the high throughput type of measurements that we perform. So there can be a plethora of different types of protocols, and basically it depends on the complexity and duration of the protocols the users can select, and also, let's say, the practical application referring to a given experimental setup or a given experimental question. Another point that is important to mention is the duration of the protocols. So when speaking about, let's say, not single plant level measurement, but multiple plant level measurements in this, let's say, high throughput phenotypic type of systems, there is always compromise needed in respect of how much information I gain in the time that I need to perform the measurements. And we have to basically consider the number of plants we want to phenotype. I think this will be then nicely introduced in the next talk. When speaking about hundreds of plants to be phenotyped in the meaningful time, we always need to find some good compromise. So basically here I mentioned three typical type of protocols that are mostly used. The FV over FM, which is a very simple protocol, which is fairly fast to be measured. Quenching protocol that was nicely introduced here when we can basically measure the relaxation or the quenching of the fluorescence in the light, reaching the light adapted state, and then the relaxation in the dark adapted state. Or then, here I'm mentioning that this type of a protocol, let's say, the fastest setup almost we can do for this type of protocol is in the range of 2.5 minutes, whereas for FV over FM, we need basically just a few seconds to perform the measurements. In using protocols as light response curve, so measuring the photosynthetic efficiency in different light intensities, there we always need a time for the light adaptation of the plants, and we basically reaching, let's say, semi-light adapted states. And there we have basically, we are in a range of number of minutes for performing such type of a protocol. An important thing to say, for all these protocols, we need dark adaptation. And Tracy briefly touched the question of dark adaptation. I would like to get back to this because this is a very common question and lots of discussions actually. What if to dark adapt and how long? So yes, when to dark adapt, when we need the F0, so the minimum level and maximum level of fluorescence be determined, primarily for NPQ measurements and calculations referring to non-photochemical quenching. parameters. How to perform the dark adaptation. In the non-imaging tools, we had different types of reef clips that can be used for shielding from ambient light. Or we can do dark adaptation of the plant samples in, let's say, a dark room environment. In the high throughput or in the imaging type of systems, we can use a dark adaptation tunnel or dark adaptation room. Usually safe green light is useful to be used for the safe operation of the material. Because we always need to see at least something. And how in the field to dark adapt the samples, or also in, let's say, the high throughput type of setups, we can use a natural night adaptation, where we can use a pre-dawn type of measurements, let's say in about, usually speaking about measurements taken 2 hours after onset of dark. And how long the dark adaptation should be done, this really depends on multiple aspects, on the species, on the aim of the study, on the plant history as well. But out of experience, I hope I won't make a mistake when I mention that some sort of a golden standard we use and test it enough. So for Arabidopsis, that is basically low light growing plants, we use usually 5 to 10 minutes. And for most of the rest of the plants, we use 15 to 20 minutes dark adaptation. However, here it's important to say that even this type of dark adaptation, I mean, 15-20 minutes is a lot of time required, which during the day type of measurements and high throughput type of systems, one normally cannot afford. Therefore, it's useful to have protocols that can be used for light adapted type of measurements. And this was also already briefly introduced, and I would mention here some example of a short type of a light protocol where we measure the plants in the light adapted state. So we light adapt the plants, and we measure the steady state fluorescence, apply saturating pulse for determination of the maximum fluorescence level in the light, and the far red pulses for determining F0 prime values. And for the light adaptation, either we measure the plants in their ambient environment, in the field, in the light adapted state, and then perform a fast type of a measurement. Here I would mention we have basically 3 seconds of measurement for this type of a measurement. Or we use light adaptation tunnels where we can adapt the plants prior reaching the imaging station. So the plants are light adapted and then imaged exactly under the same light conditions. So that would be an example of light adapted type of measurement that, let's say, for high throughput applications, when we aim to screen the plants in a high throughput in a relatively short time span, is very useful. And what we still obtain are the key parameters as PS2 operation efficiency.
Hey Klára, can I just say the 2-minute warning maybe?
Okay, so I better speed up.
You're a little bit over, maybe.
Yeah, I think I can make it. So some important points I would like to mention and maybe remind, we heard few of them, that we always need a reference state. And so basically having the photosynthetic organism in a well-defined steady state. This is either dark adapted or a light adapted sample. I want to mention that for the measurements during the day, also what matters is the time of the day, because fluorescence yields is circadian regulated or is under circadian rhythm regulation. So this is also important to mention. And then the protocol selection is the key feature that one needs to define and decide what type of protocol should be measured. And what type of parameters finally the user can obtain. Referring to hardware and protocol settings, the key for chlorophyll fluorescence imaging measurement is a homogeneous light illumination over the defined imaging area. And also, that's maybe I would mention here, that's why if you look at this example, you see that the imaging panel, here we have in the middle the imaging camera, and then we have illumination LEDs. You can see that the imaging panel is 1/3 larger than the object that is being imaged. That's to ensure that the entire area that is where the plants are defined is homogeneously illuminated. Another important aspect is that there is a minimal change in the light intensity with the distance increase from the plant object. And then few more aspects. The measuring pulse should be short enough not to result in an actinic effect. Actinic effect meaning that the F0 value is not F0, but basically gradually increasing to FD value. We need the suitable light intensity for the actinic light, referring to the cultivation light intensity. Then we need a strong illumination saturating pulse, and often the question is, how do I define the intensity of the saturating pulse? And here I think, I hope easy advice can be used, that it's usually 5 to 6 times of the actinic light intensity. And then also additional lights are important, as was already mentioned, the far-red lights for F0 prime measurement. And for the applications of chlorophyll fluorescence, I will try to be effective now. Briefly mentioned by Tracy that we can use it very well actually for looking at the details of spatial heterogeneity. Here, photosynthetic efficiency in the leaf treated with DCMU, when PTO was submerged to this DCMU. Screening, where we can use steady state signal of the plants in the large scale screening for actually mutants that clearly differentiate in the fluorescent signal or the given photosynthetic parameter, here maximum quantum yield. And the imaging can be used very effectively really for high throughput, high scale type of screening. Here example from IPK, where Arabidopsis or 384 Arabidopsis accessions were screened for their PS2 operating efficiency. And to demonstrate that the same type of a system can be applied also for large plants, the same was performed for a large collection of maize accessions, and the PS2 operating efficiency was screened also in, let's say, this large type of plants. Further, I would like to mention that we can use chlorophyll fluorescence effectively also for dynamic type of measurements, where we look at the stress response in time. And here, example from a group of Fairbank in Anno in Australia, when they use Chivas population for screening for discrimination of genes associated with NPQ. And another example, this time from the lab of Mark Tester, where chlorophyll fluorescence imaging was very effectively used to characterize early stress responses in Arabidopsis, and finally effectively was used for identification of genetic loci actually linked to this type of stress responses. And almost in the end, I would like to give you one example referring to suitable selection of parameters to be used to characterize the given stress response in the plants, and was briefly touched by Tracy about the FV/FM parameter. And here I would like to give you an example of drought stress application in Arabidopsis thaliana, when the plants were subjected to progressive drought stress, and they were kept in these conditions without watering for basically a little bit more than 2 weeks. Red is control, blue are plants that were drought stress, and we can see that basically for up to 16 days, where here on the day 18 the plants were rewatered, we don't see much effect on the level of maximum quantum yield. However, when we look at the PS2 operating efficiency of the plants, we can see that actually very early during the stress response, the PS2 operating efficiency drops in the stress plants. And when we rewater the plants, we can see a nice recovery. And we can see that even prior this response, prior the PS2 efficiency drops, the NPQ is increased in the plants as a productive mechanism. And again, actually, when the plants are rewatered, the NPQ goes back after the rewatering, and the plants recover to the same level as in the control plants. And with this example, I wanted to mention that also depends on the type of parameters one aims to measure, and it's always important to consider when designing the experiment protocol. And here, one more example, just in a different type of plants, not talking only about Arabidopsis all the time, but looking at the drought stress response in barley plants. And you can see the progression of the drought stress response here, measuring basically only a light adapted short protocol for obtaining PS2 operating efficiency. The day 17 plants rewatered, and you can see a nice recovery of the plants in the time. And one more example, which I think clearly demonstrates that the selection of the suitable parameters that one extracts from the chlorophyll fluorescence imaging really matters. And this is an example from biotic stress type of application, when grape vine leaves were treated with leaf roll associated virus. So here on the top you see the RGB images, untreated, treated plants, and on the RGB images we don't see any effect. We don't see much difference on the level of FV/FM parameter, but when we look at the PS2 operating efficiency as well, and mainly when we look at the NPQ, you can see that basically there is a dramatic change over the leaf level, and therefore like the early symptoms can be effectively detected. And not to always say that FV/FM is not the best to be used in the stress response screening. Here I have one last example that I really like to show always, because I think it's a very nice example how FV/FM can be used as a very effective also indicator of the stress. And this is a work referring to plant resistance analysis in the plants. When we measure FV/FM, you see the color scale of the plants, that false color scale for the FV/FM parameter in the plants that were treated, were infected. And we can see, we can classify the FV/FM values, threshold them, and use them for the segmentation and characterization of the different types of tissues and the symptoms of the stress response. So clearly differentiate between the symptomatic, impacted, wilted, or necrotic tissue. And this can be then classified over the time course of the stress experiment, so days after inoculation. And I hope with this I could provide enough of good examples how the chlorophyll fluorescence imaging can be applied, and I hope there is some space for discussion afterwards. Thank you for your attention.
Thank you, Klára. If I can introduce Dr. Kerstin Neumann.
Then thanks a lot for introducing me and for inviting me for this workshop. As said previously in the break, I'm definitely not a photosynthesis expert. However, I'm a user of one of the high throughput systems since long time, and since this year have overtook the group of automated plant phenotyping here at IPK after Astrid Juncker left the IPK. And we have done some studies involving photosynthetic performance in cereals in the past years by having an expert on photosynthesis here, and I want to introduce you a bit to the results that we have obtained during these high throughput experiments, and focusing here on the aspect of drought tolerance. Yeah, you see here on the image already in view into the greenhouse where one of the high throughput systems is located. Here are the imaging towers for those who have not been at IPK yet. IPK has invested substantially into phenotyping infrastructure in the past years, as this was always a bottleneck, especially in plant genetic studies dealing with screening of larger populations. And if you want to look up this infrastructure, you can go to the website, and we are part of all the relevant phenotyping networks on the national and international level. And I want to focus here on the high throughput systems located in the phytochamber and in the greenhouse, which are customized LambdaTech systems for small plant species, for medium-sized plant species, and for large plant species such as maize, cereals, or Arabidopsis. All these systems come with side view and top view imaging and with a automated watering system, and have different capacities in terms of single plants, and some have also the option putting multiple plants on one carrier and to image several plants. And since the DPPN project, all three systems got upgrades also with kinetic chlorophyll fluorescence imaging systems from PSI, and therefore I'm very thankful that we had the introduction to all these techniques from Klára before, and we have applied this also to cereals, and this is how example images look like. So it's a top view camera panel, as you have seen already in Klára's presentation, basically. This is a view to the medium-sized system, what we have used to screen the cereal collections, and it can hold up to 520 plants on single pot configuration, so one plant per pot configuration. It's 10 individual lanes, which can also randomize plants by exchanging plants from one lane to the other, and we can perform experiments definitely up to the flowering stage, but also beyond. So depending on your research question and the duration of experiments, you can screen plants even for the full life cycle, which we have done 2019 in one experiment for the first time. And this system is located in a controlled greenhouse, so you can control the temperature. You have a cooling system and a heating system. And as mentioned, you have then the imaging towers with the top view and side view cameras. And after the imaging, you have the pump balance station for watering, where you can then water by target weight or by a defined volume, depending on what you want to do. And in front, which is not visible now here, in front of the imaging towers since the DPPN project, we have this PSI camera panel installed in the first chamber of the RGB cameras, and you can move it in on demand. And in front of this imaging towers is now a light adaptation tunnel, where also you could dark adapt plants if you would want to, but usually we use it for light adaptation. And just one quick reminder why drought and heat also is such a problem for cereal and cereal breeding, or a challenge. So all the predictions by climate change predict that we also get here in Central Europe more problems with drought and heat, and we have observed that in Germany over the past 3 years by having severe drought and heat conditions in the last 3 years. And this is also one reason why the German cereal yields are stagnating over the past years, while before they had increased constantly. We had started in barley to apply a spring drought setup. As Klára has already mentioned, you can also look on the recovery in high throughput experiments, and this is actually what we have done here. So we aimed at a drought in the tillering stage, so in the vegetative stage, that is then ended as it would happen in nature when it rains again. And we looked on plant recovery in barley plant genetic resources. I will not go into detail for that because the genetics behind the biomass formation in both conditions is published here. Just that this was the base for going then also into wheat. And just one quick slide for the barley, it's how the biomass forms under both treatments. So we performed each 3 experiments with this panel. And in the well-watered conditions, of course, you have a continuous growth, which is more like reflecting a logistic growth curve. While in the spring drought treatment, in the drought period, of course, plants can still grow for a while until the drought effect is too severe and they stop growing. And depending a bit on the environment, because you have kind of seasonal effect still in the greenhouse, then even this biomass parameter from the images is decreasing, which is basically reflecting a loss of water, so a loss in fresh weight and not in dry weight. And then on re-watering, the plants are sucking up the water again and continuing to grow in the recovery period, but of course never reach the level of biomass which would be formed in the well-water treatment. And we have high data quality for all the traits coming from the image analysis, which now allows us, for example, for GWAS studies, to go with 2 replicates and 2 replicates per genotype and treatment, and 2 experiments to have a high data quality in terms of heritability. So from barley, we then moved to wheat. And when the wheat experiment started, the DPPN upgrade came, which involved this PSI panel, so the fluorochem. And this is integrated into the existing LemnaTech systems. As said, it has a top view camera panel and the adaptation tunnel. And it's more or less a semi-automatic integration. So you have to move it in on demand, and you do whether LemnaTech imaging or you do the photosynthesis measurement. And what was a challenge, of course, that we have to measure, or we want to measure 520 plants during the time course of the experiment. So as Klára said, that requires some kind of fast screening protocol. And we were lucky that Michele Greco was contacting us, and he heard about the systems and was interested, and he's a photosynthesis expert, I should say, and he was then with us developing a Humboldt grant, which he got for 2 years to develop a protocol and to apply this protocol for plant genetic studies in the ongoing experiments at this time. And he was developing a measuring protocol, and he was basing this on one of his protocols, which he had developed already before, but which was much longer, of course, because he had also just a few plants to screen at this time. And then based on this knowledge, he tested and applied or developed in the end a protocol which can be used for high throughput measurements. And that's as said in the light adapted stage. So the plants are here in the adaptation tunnel, and until they move into the first chamber where the fluorochem is, and before the light protocol is applied by the camera, they stay minimum 11 minutes, I have to say, in the high light, because the plants that come in later stay a bit longer, because you always have to add the time of movement here and of imaging in the chamber. So they have a minimum of 11 minutes adaptation to high light in this light tunnel. Then they move into the chamber where the PSI camera system is integrated. And it's a traditional parameter of photosynthesis of this efficiency of photosystem 2. which is measured then after the light pulse as 10 seconds in the same light conditions as in the light tunnel. And then this protocol continues, and Michele applies a low light to kind of mimic the natural conditions in the field, where also you have fluctuating light condition caused by shading effects. So it's only 10% of the light here, and then after 10 seconds you have another pulse, and he's measuring at this level in the transient phase. So this is in the steady state, and this is in the transient light shift phase, the efficiency of photosystem 2, and is then also looking on the ratio of these two QY or quantum yields. Moreover, he has looked at chlorophyll fluorescence just prior the saturating light pulse, and has looked also on the ratio of these two parameters here and here. And with this, we are able to measure all 520 plants in about 11 hours, including all the lane exchanges and movements that need to be done. And we were applying this in the first project, which was the EU project WILBY, which was running on the system. And these are plant genetic resources again, and we selected a spring wheat panel out of this WILBY wheat panel, which is published here by Pont et al. 127 lines, which were selected to capture a maximum of genetic diversity of worldwide origin. And they comprise land races, historic cultivars, more recent cultivars, and also breeding material and current varieties. So basically the whole breeding progress of spring wheat. And we adapted the protocol from barley by having a drought period that is a bit longer than a barley, and that is then followed by a 1-week recovery period, which is observed on the system. And then the plants were moved to another greenhouse chamber, to grow for maturity, because here we also wanted to look on the effect of the spring drought treatment on the yield components. As said before, based on the barley data, we could plan here 2 experiments and 2 replicates per genotype and treatment, and that was sufficient enough to have a high heritability of most of the traits. So it was aimed at the traits coming from image analysis. That has worked well. We also looked on the yield components. They were also quite heritable. And also the photosynthesis traits were of usable heritability, although not as high as the other traits I want to mention. How does it look in the spring wheat panel? So the biomass is similar as in barley. You have the continuous growth and well-watered condition, and you have this so-called wilting curve in the spring drought setup, followed by the recovery. Also the plant height was reduced due to the drought. And he looked here on the growth rate in the recovery period, so only in this period for biomass. And in the absolute growth rate, of course, it's higher in the well-watered plants. However, if you look on the relative growth rate, this is much higher in the drought-stressed plants, because they try to catch up with what they lost during the drought. So that's the general pattern. And we were able in these 2 experiments where he started here to measure in both experiments only at these 3 days. We have measured or tried to measure also here, but I have to say in the first experiment we had severe problems still with the integration of PSI and LemnaTech software. So we have this data only in the second experiment. So for a combined analysis, we could use only these 3 days, so the last day of drought, shortly after the plant recovery, and at the end of observed plant recovery, because of course the experiment continued outside the LemnaTech facility. Then let's have a look at the traits that he measured with his protocol. And these are here the box plots. So in blue again the well-water treatment, in orange the drought treatment. The line indicates the recovery. So here is the last day in drought for the photosynthetic efficiency under this steady state highlight condition, of course, strongly reduced in the drought-stressed plants after this long time of drought. Similar for the parameter in the transient shift to the low light, also reduced, while the ratio is increased, while for this fluorescence ratio, yeah, it was significant, but it was not as strong different in drought as in control. Then comes the plant recovery, and the parameters more or less go back to the stage of where they were before in the well water treatment, so almost come to the same values as in the well water treatment, not immediately, but at this 1 week of recovery, you barely see any, you still see some differences, but you will see later in another data set that they completely level off. So the photosynthesis was not so damaged that it could not recover anymore, or the plants were not so damaged. You also made a PCA with all the data points, and you can clearly see that here, depending on the colors, so the blue colors are the 3 days in well-watered conditions, and you have red for the last day in drought, orange for shortly after plant recovery, and green after 1 week of recovery. That yeah, the severe drought stress plants cluster here. Whoops. Then the photosynthesis data move more to the well-watered and plant recovery data, which are basically clustering at the same angle. So that shows that the photosynthesis switches back to the normal state. We also said, we also looked on the yield components, and this is what you see here. So these are now the data at maturity, so plant height at maturity, biomass, straw weight, harvest index, grains per ear. We looked on total grain number, on the ear number, and so on. And maybe you see that here better. So here we checked the average trait reduction caused by drought, and there is barely an effect on 1,000 kernel weight and its components, because also the drought was happening before the flowering time. There is a stronger reduction also already in the tillering, so in the ears that formed on the spikes, that had an effect on plant height, harvest index, and so on. However, the strongest effects were observed for the grain weight of the plants, which was reduced by 65% compared to control, and mainly caused by a loss in grain number and grains per year, while the straw biomass and the total biomass was not as strong reduced compared to the grain weight. So it was a severe drought scenario. We then looked on the correlation of photosynthesis to the yield-related traits, and that's here this plots first for the well-watered conditions. You have here day 50, which is would be the last day in drought, but in well-watered, of course, you don't have it. Day 52 and day 57. And what you see here is the growth rate, absolute and relative in this recovery phase, the final biomass and the final grain weight. And here you have the QYH, QYL, the ratio, and this FS ratio. And we see to the biomass that there is a correlation of QYL and the ratio to the final biomass, but there's no, here the last column, there's no correlation of photosynthesis traits, despite even a slightly negative one here for QYH to grain yield. While this changes under drought, that's here now important, the timing. So that is the last day of drought. There's no correlation to grain yield. There is some correlation to biomass, but this is opposite here. Here's a negative effect, here a positive for these two traits. While then after the plant recovery, you have a positive correlation to grain yield and also to the biomass. And this is for the QYH, so under highlight and low light, they are positively correlated with the final biomass, while the ratios are positively correlated to grain yield in this drought-stressed plant in the recovery phase. Moreover, I told you we had a diverse collection here with different breeding history. And we also looked on kind of breeding progress in the well-watered treatment. As expected, you see the influence on plant height, which was reduced by breeding. We also got some reduction in the final biomass in this panel. And we now wanted to see how this is in the photosynthesis traits. And these are here now the 3 days for QYH. This is all in the well-water treatment. and there is a positive trend or increase along the year of release. So here you have the year of release, I should say. So there's a positive trend of breeding for QYH, not always very strong, but significant here, and in trend visible here. And there is a decrease in the QYL and in the ratio, and again an increase in this fluorescence ratio, low to highlight. And this is basically summed here. What we also see, despite that there are these significant trends, is that this might be a negative effect of breeding on the capacity of the photosynthesis activity to maintain the efficiency of the machinery from this shift from high to low light. However, we also see a large diversity in the groups, so in the subpopulations here, the landrace and so on. So that would suggest that this was rather an unintentional selection, probably, and not linked to the main traits which were under selection. And that leaves, again, room for breeding for this trait. We have then also looked on the GWAS in this data set. So these were exome capture data, so more than 450,000 informative SNPs in this data set. And I can't go into details into all the QTLs we found. Just in total, we found a number of 1,854 QTL for all these traits and time points. And Michele classified them into occurring during the pre-treatment period, so before the drought starts, then QTL appearing after the treatment started, only in the control treatment or only in the drought period treatment. And we also looked on co-localization between traits, and basically here he classified QTL regions that are having QTL for photosynthesis-related traits, and then looked also for co-localization to the other traits at maturity, and also for the vegetative image-based traits. And in total, we found 122 such regions, from which 77 were co-locating with the further traits, which you see here in the Venn diagram. So, of course, as expected, there's the largest overlap between photosynthesis and biomass QTL. However, you also see 52 of these QTL regions only valid for photosynthesis. And then you have here, all the combination between the traits. So there is substantial overlap between QTL for photosynthesis and yield and biomass related traits. We then moved on to winter wheat, which is a different panel. It's also comprising the breeding progress in 220 winter wheat varieties, however, of more recent origin. So the earliest or the oldest one are from the 1960s, 70s. And they go up to 2013, when the Brevex project basically was written. And they are mainly from Central Europe, so relevant breeding germplasm. And the breeding progress with all the field experiment is published 2019 here by Vosfeld et al. While we had the part to screen this collection on the LamnaTex system. And of course, winter wheat requires a vernalization step, and we were then transplanting the vernalized plants to these pots, put them then on the LamnaTex system, applying the spring drought setup, which also looked similar as in barley and in the spring wheat panel. So here again, the drought stress period, here the recovery period, and as in the Wilby, moved them to grow to maturity. And you see the biomass loss over time here, and you see also the reduction in the yield components. The drought was not as strong as in Wilby panel, so the yield reduction are a bit less. However, it's also the grain yield which is affected most. And in these experiments, we could look on the photosynthetic efficiency as well, using the protocol Michele had developed. And here we were able to measure the full curve, starting from even a day before the drought starts, along the drought stress period. Here at the rewatering point had also some more measurements in this period. And you see basically now that the photosynthesis of both QYL and H is going down, and quickly depending a bit on the parameter, recovering in the rewatering phase. Yeah, I said the heritability of the photosynthesis is a bit lower than the normal traits. However, most of the time point trait combinations are of decent heritability to be used for GWAS. What's maybe also interesting is the treatment correlation. So that's here the correlation coefficient over time between QYH in control and stress condition and between QYL in control and stress condition. As you see, there's some higher treatment correlation for the steady state photosynthetic efficiency compared to this transient value or measured in this light shift. And it's decreasing in both for both parameters with a progressing drought and in the recovery phase. Yeah, recovering and but still you have room for genotypic variance, so it's not a super high correlation, leaving room that different cultivars are performing good under drought compared to control condition in this phase. Yeah, this we already said. Moreover, we also have the yield components in this data set, and it's just one example of the correlation of QY, H and L under control and stress condition to these parameters measured at maturity, and this QY at day 50, which is the end of recovery. And you see there's some correlation, not for all traits, with the photosynthesis, especially here for biomass, and that's only valid for the stress treatment. So that's QYH under drought stress in the recovery phase and QYL under drought stress, while the control treatment is not correlated with biomass. For grain yield here in this data set, it's QYH under stress, and there's also a correlation to the straw yield. Interestingly, the QYL parameter of both control and stress treatment here is negatively correlated with the harvest index, indicating this is really more for the straw biomass growth, while the grain yield model probably depends on the QYH. And there's also a positive correlation between this parameter and the 1000 kernel weight, while these correlations are absent in the control treatment. And also here we were looking then on the breeding influence of photosynthesis in this elite winter wheat panel. And that's here again the timeline, days since the start of drought, and then you have the plant recovery. And you have here now the correlation coefficient to the year of the release and each of these traits. So QYH under well-watered, QYL under well-watered, under drought stress conditions, the hedged bars. And you see, especially for the QYH parameter, there's some higher correlation with the year of release. So as in the wild bee panel, this was increased by breeding, so it's a positive effect, while for the QYL parameter, yeah, the correlations are not as high. There are some where you have a positive trend, there are also some where you have a negative trend, but this is not even significant. So let's say in control, you can say QYH was increased by breeding in this panel. For QYL, you get an unstable picture. Under drought, you see this breeding effect reducing, but it comes back in the recovery phase. So here these bars get smaller while they come back under recovery. While you also now see a breeding effect of QYL under drought, which is then also at a much lower level present in the recovery phase. And if we look here in the well water treatment during this period, we had also tested then correlations to field yield data from the BLIVEX consortium, from the Phosphels et al. Data set. And we got positive correlation between QYL in the well water treatment and field grain yield, and it was strongest at this time point. So the next slide is basically the last slide with data that I show. This is this parameter at days after transfer, is it now here 30, with a grain yield field, so from the field, with the blues over 3 years of multiple location field data. And this is explaining 21% of the grain yield to photosynthesis And it gets even more prominent if you here have the photosynthesis value of the lowest, or the 10 genotypes with the lowest grain yield compared to the 10 genotypes with the highest grain yield. So there's definitely a larger difference in the photosynthetic efficiency for these genotypes. And yeah, with that, I want to summarize a bit what we have found so far. Of course, we are still checking and analyzing and comparing. So Michele's adapted protocol allows for high throughput screening of plants under control and stress conditions. We have applied this to drought, but we already have applied this as well to heat stress, and that also worked well and shows differences between the treatments and genotypes. This measurement, or this additional measurement in this light transition phase from high to low light, probably better reflects what happens in nature, where you also have fluctuating light conditions. And we also clearly see that these two efficiencies are not the same. They are not or poorly correlated in most of the time point treatments combinations. What we also see that there is a full recovery of photosynthesis after the drought stress period. However, in the collections, the treatment correlations are low to medium, so the genotype ranking is changing, and thereby also the genetic architecture. What we also see is that under drought, you have a higher relative growth rate in the recovery because the plant tries to, or the genotype tries to catch up with what was lost. And then there's in this phase a correlation between photosynthesis traits and the final biomass and yield, which might reflect an increase of zinc demand during this plant recovery phase, where growth is potentially limited by the photosynthesis, which is not the case in the well water treatment, and therefore these correlations are absent or lower. In the Wilby panel, we also saw that QYL, which was stronger correlated to the biomass data and accumulation than QYH under constant light. That was in the Webex panel, the elite winter wheat panel, not the case. And we saw, however, in both panels a potential breeding effect on QYH in an increase, while in the Wilby panel we saw for QYL a decrease. And in the Webex panel, which I just showed you, we saw a potential connection also of photosynthetic efficiency in the vegetative stage to the grain yield in the field. But of course, that still needs to be explored more in detail. And what we found in all the GWAS studies is that the genetic architecture of photosynthesis is very complex. You find a lot of QTLs. And as expected, of course, we also find often co-localization with the biomass and yield-related traits. Yeah, and with that, I finished my presentation. And of course, I have to thank all the people involved in conducting the measurements, discussing the scientific results, conducting the quantitative statistics, and so on, the image analysis. And I want to thank especially Michele for his great work here. And it's a pity that you could not be here today. And I want to thank all of you for your attention.